Anita Mishra from Biyani Girls College. Today I am going to deliver a video lecture on cohesin and condensins on the behalf of Kppo.com. Now we will going to discuss cohesin and condensins. Actually, what are cohesins and condensins? Cohesin and condensins are the molecular cross linkers. We also call them molecular glues because they are attach the molecules together during the cell cycle and cell division. Okay. There are two types of molecular cross linkers. One is the intermolecular cross linker and one is the intramolecular cross linker. Intermolecular cross linkers hold the different molecules together, and intramolecular cross linkers are present within the molecules. They hold the same molecules together. Now, intermolecular cross linkers or cohesins, their function is to hold the sister chromatids together during DNA replication. When the DNA is doubled during cell cycle, during DNA replication, these cohesins hold the two sister chromatids together during DNA replications until the cell is ready for the cell division. Actually, the chromosomes are separated in the anaphase of the mitosis or meiosis. Till that stage, this DNA is held together by the intermolecular cross linkers called cohesins. Okay, these are the cohesins which are linking together the two chromatids. Okay, now these are the intermolecular cross linkers or condensins. What are condensins? They cause the condensation or compaction. They promote the chromosome compaction by interlinking the same segments of same chromatid. Okay. Now, what is the function of these condensins? These condensins condense the chromatin material and chromatids during cell division, and by condensation they cause the condensation of the chromosomes, which facilitates the separation during cell division. When the chromosomes are moving apart from the metaphase plate towards the poles during cell division, when the chromosomes are separating. Okay. Now, these cohesins hold the two chromatids together. Now, these chromatids should be removed during anaphase when the cell has entered into the metaphase, and then when the cell will enter into the anaphase, these cohesins should be removed from the chromatids for the separation of the chromosomes. Okay. And for this there is a separate protein called saparase or saparin, which is a protease. Actually, it is a cysteine rich protease which cleaves the protein cohesins. Okay. This cohesin is a multi-subunit complex, and this saparin or saparase will cleave the cohesin at subunit SCC. Okay. This SCC subunit will be cleaved and the saparase will be released. This saparase will separate the cohesin subunits, so that cohesin will now no longer will be the functional unit and will allow the chromosomes to be separated. These are the subunits of the cohesin. There are two SMC proteins. Okay. And these two SMC proteins are SMC1 and SMC3. What is the full form of SMC? Structural maintenance of chromosomes. These are the two subunits, and the third subunit is alpha cleasin, and the next subunits are STG1, 2, 3. The SMC proteins are actually the ATPases. They help in providing the ATP hydrolysis, and they provide the energy for the function of the cohesins. Alpha cleasin is attached near the centromere. It facilitates the attachment near the centromere, so that the chromatids are attached at the centromere position. STAG1 and 3 are present on the chromosome arms. They facilitate the attachment at the chromosome arms. Now, these condensins, which are also promoting the cell division, they condense the chromosomes, and their maximal activity is during mitosis and meiosis, because they are facilitating the chromosome separation. But they are down regulated in the interphase by some of the proteins they are down regulated because they should not be active during interphase, and chromosomes should not be condensed during interphase. It should be present like a chromatin material, and by some of the proteins their activity is activated during mitosis and meiosis, so that they are activated and they will promote the cell division. These cohesins are also of two types, first and second, and the similarity between them is that they both contain the core subunits SMC2 and SMC3. These SMC2 and 3 are ATPases which are providing ATP for the activity of these condensins and cohesins. And so, this is all how the cohesins and condensins helps in separation of the chromosomes during cell division, and how their activity is regulated. Thanks. I hope that you have understood the cohesin and condensins functions and their structures. Thanks for watching my video.